Good afternoon, everyone. Today, we embark on a journey to demystify the intricacy of past year exam questions for May 2023 for A-level students. Examining the past offers us a compass to navigate the challenges ahead, empowering us with the insight and strategies to conquer the present. Let's delve into the treasure trove of the past exam, unlocking the key of success and mastering the art of academic excellence. Question number 1. The electron micrograph showed onion root cells prepared using the freeze fraction technique. The cells were quickly frozen and then physically broken apart. Freeze fracture breaks apart cells along with areas such as membrane and the surface of organelle. Which statement best explains the experience of the electron micrograph? Based on the freeze fracture technique, porosity to break cells along with areas such as membranes and organelle surfaces, the appearance of the electron micrograph suggests that the cells were broken apart at the nuclear envelope with structure X being a nucleopore. The answer will be B. Which cell structures can form vesicle? Cell surface membrane is formed vesicle through processes like endocytosis and exocytosis, and endoplasmic reticulum form vesicle involved in protein transport and lipid metabolism. And Golgi bodies form vesicle that transport molecules to different parts of the cells or to the cell surfaces. The answer will be cell surface membrane, endoplasmic reticulum, and Golgi body. These are the cells can form vesicles. Number 3, 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. First, synthesis of polypeptides. 2, synthesis of lipids. And 3, packaging of hydrolytic enzyme that will remain in the cells. The appearance were listed by letter. V is actually represent membrane which surround and enclose inner cavity. W represent non-membrane bound spherical structures. X is a double membrane bound cylindrical structures. And Z is a membrane bound sets arranged as a flattened stack. Which student correctly match the number functions with the appearance of the cell structure? The answer will be W, X, and Z, which is not belongs to any answer of given. Synthesis of polypeptide W is a non-membrane bound spherical structure. Synthesis of lipid X is a double membrane intersphere plus pause, and packaging of hydrolytic enzyme that will remain in the cells. Z membrane bound sets arranged as a flattened stack. Number 4, which cells contain a tonoplast? The answer will be the tonoplast is a membrane that surrounds the vacuole in the plant cells. Among the options provided, the cells that contain a tonoplast are root hairs, sieve tube element, and endodermis. The answer will be B. Which organelle found in animal or plant cells are surrounded by double membrane? The organelles surrounded by double membrane in both animal and plant cells are mitochondria and nucleus. So the correct options is B, which is chloroplast, mitochondria, and nucleus. Some scientists think that mitochondria evolved from bacteria that entered the cytoplasm of different cells and were able to survive there. Which structural features of mitochondria support this hypothesis? The structural features that support the hypothesis that mitochondria evolved from bacteria are the folded internal membrane, circular DNA, and 70S ribosomes. The answer will be A. What is present in all viruses, all prokaryotes, and all eukaryotes? Cytosine is a nucleotide basis found in DNA and RNA, which are present in all viruses, prokaryotes, and eukaryotes. The answer will be cytosine. The table shows some steps that can be made in carrying out the benefit test. Which combination of steps is required to carry out a semi-quantitative test on a reducing sugar solution? First is a standardized volume of benefit solution and volume of test solution, and then followed by standardized boiling time with benefit solution, and compare final color with numbered color standard. Number 9, the diagram shows 3 examples of different bonds. Which bond hold the secondary structure of protein together? So the bond that hold the secondary structure of protein together are option bond 1, which is NH, O equals to C is hydrogen bond. This bond formed between the amino hydrogen NH and the carbonyl oxygen, which is O equals to C of the peptide backbone. The answer will be number 1, which is bond 1 only. Insulin is a globular protein involved in a cell signaling. It is transported in the blood plasma from the cells that synthesize it to its target cells. A molecule of insulin contains 6 sulfur-containing amino acids and has 2 polypeptide chains. Which statement about insulin are correct? Amino acids with hydrophobic R group will be found in the center of an insulin molecule. Insulin is a globular protein, and hydrophobic amino acids typically reside in the interior of the protein structure, away from the surrounding aqueous environment. So the answer will be an insulin molecule has a quaternary structure, and amino acid with hydrophobic R group will be found in the center of insulin molecule. Number 11, which statement correct explain why blood plasma can be maintained at a stable temperature? High specific heat capacity means that blood plasma can absorb and retain a significant amount of heat without undergoing a large temperature change, which helps to stabilize its temperature. The answer for this will be, it has a high specific heat capacity. Which region on the graph show the activation energy of an enzyme catalyzed reaction? The answer will be B. The graph shows the effect of an increasing substrate concentration on the rate of an enzyme catalyzed reaction. Line P represents the result when the enzyme is used at its optimum pH and optimum temperature and without an inhibitor. Line Q represents the result when the reaction conditions are changed. Which description of changes to the reaction condition could result in line Q if all other conditions were kept the same? The answer will be add an inhibitor that has a similar shape to the substrate and add an inhibitor that blocks the active site of the enzyme. Number 14, which statement about cell signaling is correct? The binding of a ligand may cause a change to the shape of the receptor. For students A, B, C, and D observed plant epidermal cells that had been placed in a concentrated sucrose solution for 30 minutes. They were asked to identify the partially permeable layer and to explain the appearance of the cell in terms of water potential and movement of water. Which student is correct? Partially permeable layer will be cell membrane, cell surface membrane. Water potential at start of experiment is a cell content have a higher water potential than the sucrose solution, and movement of water during experiment will be more water moved out of a cell than moved in. The table compares the surface area to volume ratio of the 5 agar blocks that differ in dimension, but which all have the same volume. The agar block can be used to measure the efficiency of diffusion, where efficiency is measured at the time taken for a dye to reach all parts of the block. Which prediction can be made about the way in which size and dimension of this block affect the efficiency of diffusion? The answer will be C. The efficiency of diffusion will increase as a block of fixed volume is flattened. 16. The cell cycle include mitosis. Which are features of this type of nuclear division? Form cells of equal size to the parent cells, which is number 1, is correct because mitosis results in the formation of two daughter cells that are usually identical in size to the parent cell. Number 2, form genetically identical nuclei is also correct because mitosis ensures that each daughter cells receive an identical copy of the genetic material, resulting in genetically identical nuclei. And number 3 is incorrect because semi-conservative replication of DNA occurs during the S phase of the cell cycle, which precedes mitosis. During mitosis, the replicated chromosomes are segregated into daughter cells, ensuring each cell receives a complete set of chromosomes. So the answer would be 1 and 2. A student observed the cells in the growing region of an option of an onion root and obtain the data shown, and which percentage of cells contain chromosomes that appear as the two chromatids. So below is the calculations how you divide the percentage of 8.9%. So to determine the percentages of cells containing chromosomes that appear as two chromatids, we need to focus on the stage of interphase, since during interphase, chromosomes are replicated, resulting in each chromosome consisting of two sister chromatids. So the total number of cells in interphase is about 886. Percentage of cell in interphase is equal to number of cells in interphase divided by total number of cells multiplied by 100 and divided by 10. And you get the value of 8.86%. So approximately 8.86% of cells contain chromosomes that appear as two chromatids. So when you round the value, they get 8.9%. The answer will be B. 19. Which statement about messenger RNA is correct? In eukaryotic cell, mRNA is synthesis through RNA processing, which involves the removal of intron, not exon, from the primary RNA transcript. B is also incorrect because while mRNA is a single-stranded and contains different nitrogenous bases than DNA, it contains the same purine bases as DNA, adenine and guanine. And C is correct because mRNA molecule contains ribose sugar joined to phosphate group by phosphodiester bond. And D is incorrect because the monomer of mRNA contains ribose sugar, not deoxyribose sugar, joined to a phosphate group and a nitrogenous base. The answer will be on the number 19 is C. Number 20, which structure are involved in transcription only? The answer will be DNA template strand and RNA polymerase. One gene provides the code for the production of which type of molecule. Genes contain the instruction to produce protein, which are made up of change of amino acid called polypeptides. Each gene provides the sequence of amino acid that make up a specific polypeptide change. So the answer will be polypeptide. Number 22, the table shows some mRNA codon that code for certain amino acid. A DNA template strand has a base sequence shown ACA, GTA, TTA, TTT, GCAA, CG. What would the change in the amino acid be if the first base in the 50NA triplet was a substitute of NA base? So the answer will be IG9 to side cycling. Number 23, which structure contains cytoplasm with mitochondria and nucleus? The answer will be companion cells, which is A. What is the correct term to describe intermolecular hydrogen bonding between water molecules? This would be cohesion. 25th, the diagram shows the outline of the xerophytic sleeve that had been left for 55 minutes in different conditions P and Q. Which statements about the cells in layer Y of the leaf in each of the conditions P and Q after 45 minutes are correct? So the answer will be there is a less negative water potential in P than in Q, and the cells may be turgid in P and plasmolized in Q. And number 4, there is no net diffusion of water into Y in either P or Q. 26, how does sucrose move from chloroplast to a folium? Is through 3 different processes: diffusion, apoplast pathway, and symplast pathway. Sucrose can indeed move through all 3 pathways: diffusion, apoplast pathway, and symplast pathway. However, the symplast pathway is a primary route for sucrose movement from the chloroplast to the folium. How are companion cells involved in loading sucrose into folium sieve tube element? Companion cells active load sucrose into folium sieve tube element using contrast-spotted protein. This active transport mechanism, which is ensure efficient movement of sucrose from the companion cells into the sieve tube element, which then transport it throughout the plant. Additionally, there is also passive movement of sucrose through plasmodesmata, which are channel connecting the companion cells and sieve tube elements, aiding in the overall transport process. Therefore, both active co-transporter protein and passive plasmodesmata mechanisms are involved in loading sucrose into folium sieve tube element. The answer will be A, actively through co-transporter protein and passively through plasmodesmata. The photomicrograph shows blood cells as seen using a high-power light microscope, which row correctly identify the different types of white blood cells. Number 1 is actually represent neutrophil, and number 2 is monocyte, and number 3 is lymphocyte. Plus, plain diagram of 2 blood vessels are shown, which labels are correct. Number 1 is artery, number 2 is vein, number 3 is collagen fiber, number 4 is smooth muscles and elastic tissue, and number 5 is endothelium. What are found in the blood and tissue fluid? Carbon dioxide found in both blood and tissue fluid, as it is a waste product of cellular metabolism that needs to be transported from tissue to the lung for exhalation. Fatty acids also found in the blood as they are transported bound to protein as lipoprotein, but are less abundant in tissue fluid. And the white blood cells mainly found in the blood as part of the immune system defense mechanism, although small numbers may mitigate into tissue fluid during inflammatory responses. And protein also found in the blood, in both blood and tissue fluid, but the types and concentration may vary. Blood contains a wide range of protein, including albumin, globulins, and clotting factors, whereas tissue fluid contain protein that have leaked from blood capillaries. So, all options are found in blood, but not all are equally present in tissue fluid. So, the answer will be 1, 2, 3, and 4. Which reaction could be slowed down by the inhibitor of carbonic hydroxide? The answer will be 2 and 3. This is the answer. 2 and 3 is given by the Cambridge, but the actual answer is number 2 only, which is not listed in the answer. An inhibitor of carbonic hydroxide would slow down reaction 2, which is the conversion of carbon dioxide and water into carbonic acid. Carbonic hydroxide catalyzes this reaction, so inhibiting it would impede the formation of carbonic acid. Therefore, the correct options is CO2 plus H2O to produce H2CO3. The answer only number 2. 32. Which sequence of letters correctly identify the order of event during the cardiac cycle? First, the correct sequence of event during the cardiac cycle is as follows. First is X, which is wave of excitation spread from the sinoatrial node. And then followed by T is atrial wall contract. V if a wave of excitation enters the anteroventricular node. U is impulse is delayed by a fraction of a second. W if a wave of excitation passes down the Purkinje fiber. And Y is a ventricle contract. So the correct sequence of letters is X, T, V, U, W, and Y. The answer will be C. Number 33, the diagram gives information about blood pressure in the left side of the heart during the cardiac cycle. And wave open and close at the point number. Which row identify the valve opening or closing at the point number? Anteroventricular wave opens, number 4. Semilunar wave open, number 2. Anteroventricular wave closes, number 1. And semilunar wave closes, number 3. The answer will be D. A person with no breathing condition rests for an hour. Their breathing in this time is shallow and slow, so little air from outside the body reaches the alveoli. The person's heart rate remains constant. Which statement is correct? When a person is at rest and their breathing is shallow and slow, the rate of oxygen molecule diffusing from the air in the alveoli into the blood is slower compared to when the person is active. This is because during physical activity, the body's oxygen demand increases, leading to deeper and faster breathing, which in turn enhances the rate of oxygen diffusion into the blood. So during rest, with shallow and slow breathing, less air reaches the alveoli, resulting in a slower rate of oxygen diffusion. The answer will be B. Which statement about all bronchioles are correct? They are epithelium. Bronchioles are lined with epithelial cells which form the inner lining of the airway. They have goblet cells. Goblet cells which secrete mucus are present in the epithelium of larger bronchioles and play a role in airway defense. And number 3, they have muscle tissue. Bronchioles are surrounded by smooth muscle tissue which allow them to contract or dilate, regulating airflow to the alveoli. The answer will be 1, 2, and 3. 36. The diagram shows some of the pathogens that cause disease in humans and some of the way they are transmitted. Which is the correct pathogen and methods of transmission for malaria? The answer will be 2 and X. 2 is a protoctis and X is a mosquito bite. 37. Some of the processes which result in the formation of a population of bacteria that are resistant to a new antibiotic are listed. 1 is a change in reproductive success of bacteria. 2, increase in frequency of the resistant alive in the population. 3, increase in genetic variation within the population. And 4 is a random mutation occurs in bacterial DNA. What is the correct order of these processes? First is actually random mutation occurs in bacterial DNA. Random mutation in bacterial DNA generate genetic variation, including mutation that confer antibiotic resistance. Number 3, it increase in genetic variation within the population. Mechanisms such as horizontal gene transfer and genetic recombination increase genetic variation within the bacterial population. Number 2 is increase in frequency of the resistant alive in the population. Bacteria carrying alleles for antibiotic resistance have a higher chance of survival and reproduction, leading to an increase in the frequency of this allele within the population over time. And number 1, it will change in reproductive success of bacteria. Bacteria with antibiotic resistant traits have a reproductive advantage in the presence of the antibiotic, leading to the increased proliferation and dominance within the population. So the answer would be 4, 3, 2, and 1, which is not listed in any answer. It's either A, B, C, or D. So the correct answer would be 4, 3, 2, and 1. 38. When bacteria are grown in a petri dish containing these with antibiotics, there will be a zone of inhibition of bacterial growth. The chart shows the size of the zone of inhibition when a species of bacteria was incubated on 5 different plates of agar, each containing a dish with a different antibiotic. And which conclusion can be made about the most and least effective antibiotics on this species of bacteria? So the answer will be most effective antibiotic, which is 3, and the least effective antibiotic, which is 2. Which blood cells type does not recognize, engulf, and digest non-cell particles? The blood cell type that does not recognize, engulf, and digest non-cell particles is T killer cells. T killer cells, also known as cytotoxic T cells, are a type of lymphocyte which is involved in cell-mediated immunity. They primarily recognize and destroy infected or abnormal host cells, such as virus-infected cells or cancer cells, through the release of cytotoxic molecules. They do not typically recognize and engulf extracellular non-cell particles like macrophages, neutrophils, and other phagocytes too. The answer will be T killer cells. Repeated infection with malaria result in more effective immune to malaria. Which type of immunity is responsible for the more effective immunity? Natural active immunity occurs when the body's own immune system responds to an infection such as malaria by producing antibodies and memory cells. With repeated exposure to the malaria parasite, the immune system becomes more efficient at recognizing and fighting off the infection, leading to a stronger and more effective immune response. This is why natural active immunity is responsible for the increased effectiveness of the immune response to malaria with repeated infection. So the answer for 40, which type of immunity is responsible for the more effective immunity? It will be the natural active immunity. The answer will be C. That's all for today's presentation. Thank you very much for your cooperation. And please subscribe to Jom Stabila YouTube channel. Hope you are doing very well in your studies. Thank you very much.